Very good morning, my students. So after finishing our jelly electrophoresis in our last lecture, now we have collected our respective different DNA fragments of different sizes, right? So the process which we are going to discuss that is important for every aspect of either it's a objective or it's a subjective part. Now, polymerase. Today we will actually talk about the process which is these days used in practically like to find out many diseases, right? This is called polymerase chain reaction. We got our fragments. Now we are going to amplify our gene of interest. We are going to increase the copy number of our gene of interest, or you can say we are increasing the number of our DNA fragment. So to increase the number, to increase the copy number, there the process which is used, this is process is known as polymerase chain reaction. This is also known as gene amplification, or it is also known as amplification of gene of interest. Using the technology, which technology we are going to use today? That is polymerase chain reaction. Very well advanced technology, which many times, very frequently these days used in biotechnology department. Now, this technology, it was developed by scientist Carrie B. Mullis. The scientist name is important for us, so please highlight it. Carrie B. Mullis, he developed this technique. And what is the purpose of this technique? What is the main purpose, do you think? I told you in the starting also that we are going to use, this is used specifically to amplify, to increase the number of your DNA fragment. So it increase the number of single DNA fragments. So if you are going to increase the number, then definitely you will get lots of copies of DNA fragment. The principle on which it works on, it is a very temperature sensitive process, and in this whole process we need high temperature, low temperature, right? High temperature we will go up to 94 degrees Celsius, and then low temperature we have to maintain at least till 60 degrees Celsius. Why do we need such high temperature? All of you know that DNA is double stranded structure, right? Now, this DNA cannot be amplified in this double stranded form. So to separate DNA strand, we treat our DNA at a very high temperature. Then now we have, once these DNA strands, they are separated, we have to make the number of copies from these parent strands. For the making the number of copies, we need little low temperature. So that is why we always say that it is a temperature sensitive process. So let's quickly see the requirements we are going to need for our recipe of polymerase chain reaction. You need definitely the first thing which should be in your laboratory, that is DNA template, DNA segment. Which DNA segment, bacho, that whose copies have to be produced, whose copies have to be generated. Then you need definitely, when you say copies, वो भी from DNA. Then what is DNA made up of? DNA is made up of nucleotides. So it means you need to add more nucleotides. Who is going to help us to add those nucleotide? एक ही enzyme है बच्चों, that is DNA polymerase. But here we need this much of temperature, very high temperature for adding our nucleotides. So for that we used a very thermostable polymerase which is known as Taq DNA polymerase. Then you need primers, and we know that polymerases enzymes, they work from 5' to 3' direction. So suppose if your parent strand is 5' to 3', here you will start adding this. First you will add primers at the 3' end of the template, and the second strand is, if suppose 3' to 5', then you will add primer here at the 3' end of your template strand. So you will add primers. Another thing, definitely now you need to add more and more nucleotides. So you need activated deoxyribose nucleotide triphosphate, right? Nucleotides. You need deoxyribonucleotides, as well as some process, some mechanism, some settings to increase and decrease the temperature. Right, so I think recipes, our today's recipes requirement is clear to everyone. Now let's talk step by step the mechanism of PCR. PCR, it has three steps. We are going to complete it under three steps: denaturation, annealing, and extension, right? Denaturation means if our strand is double stranded DNA, then we are going to separate these two strands. So DNA segment which is to be amplified. Those DNA, that double-stranded DNA which has to be amplified, it is heated at a 94 degrees Celsius temperature. And once you heat your DNA at such a high temperature, there will be separation of two strands. Now, what will you get? Your strands are separated out, and each strand will act as now template for the synthesis of new DNA strands, right? This is the denaturation. The temperature is 94. Then is the annealing. Now, these are your separated strands, and I told you that if suppose this is 5 dash to 3 dash, this is 3 dash to 5 dash, you will always add your small sequences of nucleotides towards the 3 dash end of template. So you will add now primers. These small nucleotide sequences, they are known as primers, and they are always added towards 3 dash end of template strand. So a primer is what? It is a oligonucleotide 10 to 18 nucleotide sequences which is added at the 3 dash end. And now for this step, you don't need a temperature like 94 degrees Celsius. You need a little low temperature, that is 50 degrees Celsius to 65. The last step in our process is extension. It takes place definitely at a very high temperature, that is 72 degrees Celsius for this, because it is extension. Extension means you are now extending your strand. How will you extend your DNA strand? By adding nucleotides. For this thing, you need a polymerase which can sustain at a high temperature like 72 to 74 डिग्री सेल्सियस. इतने हाई टेम्परेचर पे कौन मेंटेन कर सकता है, कौन ऐड कर सकता है न्यूक्लियोटाइड? नॉर्मल डीएनए पॉलीमरेज इतने हाई टेम्परेचर पे यूजुअली इट डजंट वर्क. सो हियर for adding the nucleotide sequence, you need a thermostable DNA polymerase, and this is we obtain from thermophilic archibacterium. It is, and the name of the bacteria is Thermus aquaticus, and the enzyme is known as Taq DNA polymerase. It adds up the complementary nucleotides to 3'N, and finally it reach up to the 5'N. It completes, add all the sequences and make double stranded DNA. So every time, remember, new DNA, it will start from 3 dash end of the primer and grow किस डायरेक्शन की तरफ करता है? 5 dash की तरफ. Same here, start from 3 dash growing towards the 5 dash, right? So here is the diagrammatic representation. This was our double stranded DNA, and I have made only that segment which has to be amplified. You will heat it at 94, it will get separated, and then you will add. What will you add? You will go and add primers, right? Here you will add primers. Primers की location बहुत ज़रूरी है बच्चों. This is always added on 3 dash end of template strand. Then there will be the annealing. right? This process is your annealing when you add your uh, primers, right? And this requires your lower temperature, approximately till the 60 degrees Celsius. It can be maintained, right? And then you need that DNA polymerase plus deoxyribonucleotides. And there finally your new DNA segments is formed. And approximately if you repeat the different number of cycles, you get the millions, billions of copies of your gene of interest. So cycle is repeated again and again to obtain the desired number of your DNA segment. आपको कितना number चाहिए उसके according depend करता है. And once the DNA is amplified, this is ligated with the help of ligase to the vector DNA. And आपको पता है जब आपकी gene of interest plus vector DNA, when we combine them, they form recombinant DNA. So for tomorrow's exercise, we are going to put our DNA, recombinant DNA into the host, and once we put it into the host, we are going to culture lot of number of copies of our gene of interest at a large scale. So I will meet you tomorrow with the culture system.